All right, hello. So I will be recording how to use the eye tracker data code with MATLAB. And so I'll just be going through how to do that. So let me just share my screen. All right, so first thing you need to do is, the data from the eye tracking machine, it records into a file type called a TDA. These are TDAs. So TDAs can be opened in Excel, but MATLAB cannot read TDAs because TDAs are a weird file that is like not used by anyone else, I don't think. So you have to convert them to Excel. So all you have to do is you open up the file in Excel. You see, I have already opened it up. And then you just go to File, Save As, and then change this from this to Excel Workbook, XLSX. And you click Save, and it'll save it. Make sure you're saving it to the right file, right? So right now I have I've already converted them all, and I put them all in this folder called Excel. So these are all the TDAs converted into Excel spreadsheets. So that's step one. Step two, so the iTrack code is in this folder here. This folder, this one, my iTrack code folder has all of the files I use. So there's one main file, which is Nathan iTrack code.m. And then there are like a bunch of functions. I use a lot of functions in this code to make it easier to use. Cutter, cutter v2, full GUI, initial GUI, missing cutter, and UI pick files. They're all the functions that you need for this code. If you see there's one that's called ASV, that's a automatic save file or something. That just means that you've edited this main code and it's like a auto save before, so that you don't lose data. It's not that important for this situation. It will not be, it should not show up in your file folder, usually anyway. So it's just a bit of a tangent, but make sure all of the code is in the same folder. It's very important that they're all in the same folder. They should already be there, so just don't move them. I would avoid moving them because that'll mess things up. So once you put them onto the computer, leave them where they are. So first thing you need to do is open MATLAB. And so this is MATLAB here, this app right here. I have it on my computer here. It'll be wherever it gets downloaded to on the lab computer. So you just open it, and this is what it looks like. It will probably open up to, it opens up to whatever you had open last. So if this is the first time you're opening it, you're gonna have to open up the actual thing. Otherwise it'll just have it here automatically. So first thing you need to do is you need to open, you click open it, and you click Nathan's iTrack data code.m. You open it, and that'll open up this code. This is the script. This is the whole thing, basically. So I have some user notes, and I tried to comment it as best as I could. So if you know how to use MATLAB, free to mess with it a bit. So that's that. And then, so the other thing you need to do is you need to change the directory. So this is right now, it's looking for where I've saved it on my USB stick, but when it's on the lab computer, you're gonna have to change this directory. So how do you do that? You have to open up File Explorer. If you don't know where that is, it's this icon here. You see it, or also you can search it up, File Explorer. So this guy. And then you have to track down where the code is. So in this case, my code is here, but it might be on your desktop. It might be a folder on your desktop. But mine is right here. And you just go to where it is. So this is the folder where all the code is located. You open it. So as long as it says Nathan's iTrack code up here, and you're in the folder with all of the files, that's the right spot. So you click this up here, you click up here, see? And it'll give you the file path. That's not what I want. The file path here. So you just copy it, copy, and then you go back to the code, and you want to delete this bit here. make sure you keep the apostrophes and then you paste them in. Make sure the apostrophes are around the file path. So this basically tells the program where the functions and where the code is. So you're gonna have to do this, otherwise it will not work. So that's step one. You only need to do this the first time you do it on each computer. So if you're using it on, if you're only running this code on a live computer, you only do this once the first time. Okay. So once you have this bit fixed, all you need to do is run the code. So in order to do that, you have to go to editor, right? On the top, there's a bunch of tabs. Go to editor and you click this green run button. And it will bring up this this first dialog box. So what this is this is where you select which Excels you want to look at. So you navigate, so this goes up, right? This button will go up folders, and you can click them to open up the folder, right? So my Excels are in this folder here, Excel. And then you just select all of the people you want to do. So right now I want to run subjects 1, 2, and 3. So I can click that. If I accidentally click one, right, I accidentally add 13, I can just remove it. Just like that. So when you're done picking everyone you want to work on, click done. All right, so then this window should open up and it'll give you a lot of options. So pick the ones you want, and there's some you always have to do. So you always have to pick partial or full analysis. For this run through, I'm gonna do a partial analysis. So click partial and then for how many slides? We have 37 slides right now. As you can see, there are 37 slides, right? So you type 37, and then I'm gonna do it by slide first off. So there are two different ways you can order the slides. One is by slide set, which maintains the sequence that it's in, and there's one that is by slides, which sorts them by the slide it is. That'll make more sense once we run it. So come up with a name for it. I'm gonna call this teach ordered by slides. Done, and it'll run. And this code will save the Excel that it generates in the same folder of all of the code. So, okay, so now it's running. You can tell it's running because this says pause up here. That means it's running. And this down here says busy. It takes a little while to do it all because it has to look at all of the data and analyze it all and do all that automatic analysis for you. So it takes a little bit, but it's definitely a lot faster than having to do it by hand. It'll definitely take a little while, especially because it has to go through and find, there's a lot of things it has to do. There you go. So you know it's done when, see, this goes back to saying run, right? This doesn't say busy anymore, and your workspace here has a bunch of variables in it. That means it's done. So when it's done, you'll also be able to see so remember, this is the code folder, right? This is where the code is. It saves the Excel sheet in this folder. So it's right here, teach ordered by slides. You can open up. And here we are. So this is, as you can see, each slide. So we have Cahill Lab, light bulb, gray, rat, gray, fire rescue, the other gray, right? They're all in here. And the data is sorted out so that all of the rat slides are together, all of the light bulb slides are together, all that data is together. And hopefully this will let you get some interesting findings, right? And so here we have the subjects 1, 2, and 3. the slide just to double check. This is the Cahill Lab slide. There's the times. How long were they looking at this slide? The Cahill Lab is always going to be funky because it has calibrating, right? Average pupil dilation and the number of blinks they had when they were looking at this slide. So we have like on this light bulb slide, right? It's 10 seconds long. They blink 3, 3, 5 times. Gray, 5, 7, 6. And maybe you can find some like interesting patterns or like trends when they're looking at different slides. Sometimes you won't, but that's the scientific process, right? So that's running it for, that's ordering by slides. So that sorts out the data by the slide. So you can run all of them all at once, sort them all by slides. Now to do it by slide set, you have to do it differently because I couldn't find a way to organize it properly. So you will have to open up the eye tracking notes. So it's very important that you're keeping notes of the, when you're running the eye tracking trials. So, because one of the more important ones is it keeps track of which sequence each subject was looking at, right? So when I say it's ordered by slide set, I also mean it's ordered by sequence. And also, and we'll get into this later, but it's important to keep track of times that you've messed up recording things or like something weird is happening with the subject, like keep notes on this and like when about it happened. To make your life easier. It'll make the data analysis a lot better. But anywho, so let's look at, so okay, for sequence 2, so to organize it by slide set, you have to do it by slide set at a time because I can't sort it out automatically. So you have to figure out, so let's say we want to do the select sequence 2 people first. So that is subject 1, subject 5, and subject 9. So we'll just go back to MATLAB. We'll click run. And we will select 1. Was it 1, 5, and 9? 1, 5, and 9. 1, 5, 9. Done. And we go to partial 37, order by slide set. Call this teach ordered by slide set. Nope, I might have misclicked something there. All right, it's done. You can also tell it's done because it will say done down here. It's done. So that means it's done. We look in here and we have, here it is, teach ordered by slide set. And here we have, so this is ordered by slide set. And basically all that means is that it's slapping it all together. Like this is just, so you see this is all subject one, and this is the order. Slide set means it's in order still. So it doesn't sort it out like in slides, it's sorted out by slides. This is just keeping it all together to display all of the data. So this might be useful because if you're trying to find like weird trends, like here, see like this guy, look at this. This is all zeros. Maybe we can figure out why. This is subject, what, 9? Some sides, face slowly sinks down. So 23 and 33 might be a bit funky. 34 to the end, tracker tracks class instead of people, it's too late, right? So now we can see that here in these slides at the end, right? This is all funky because the glasses was in the way. Like these are all, this is like kind of bad. So you might not be able to use some of the data from this subject. That's why you gotta keep good notes. But you would organize it by a slide set mostly to keep the general trends together of like that person, whereas you'd order it by slides to see things for each slide. So if you wanted to see like, oh, is there any difference between the happy and the sad slides? That's why you would use slides. And if you wanted to see like, oh, like why is this one so bad? You would do by slide set because you can see general trends in the data a lot better, if that makes any sense. So that is how you do partial analysis for the ordered by slides and slide sets. And so next, I'm going to show you how to do the full analysis. So you once again click run. I'm going to do it on our slides 1, 2, 3. All right, so we have here, I'm going to do full, 37. I'm not going to name this. If you don't click anything, it will just not save in Excel. Click done. For some reason, it's going to debugging, but I will just click continue. Oh, I see. It's because I click this. There you go. That should do it. Okay, so you have two options here, RMS and moving average, and I'm going to click both right now. So the full analysis will do some filtering on the pupil dilation data. Full analysis mostly to see pupil dilation. This is where if you want graphs of the pupil dilation, do a full analysis. That's the purpose of it. And so the pupil dilation is the raw data is kind of hard to interpret. So I have two filtering options for you to see. And so one is RMS, or root mean square, and the other is moving average. So RMS, the root mean square, is here. Pull up a Wikipedia article. So it's the quadratic mean. Also, there's like, you can just read more. It's while it comes through, there's an equation for it, right? And then, and average is just average. So the way this works is that it takes, it breaks up the data by window size. So if I want window sizes of a second versus like half a second, so let's just say 0.5, I want a 0.05 window, and I want the overlap to be 90%. And let's say for my moving average, I want my window size to be like 30. It does 60 samples per second, so one sample, 60 samples per second. So 60 samples is 1 second of data, and 30 samples is half a second of data. So let's just try this, right? And I'll explain what this overlap in window size things means once it's done processing. You can see what was going on, but just gotta wait for it to do. All right. So when this is done, it'll spit out a whole bunch of graphs, and each one will show a different thing. So this here is the data for, where's the raw data? Oh, does it not print out raw data? Oh, no, it is. Wait, subject 03. Data, oh, and we see, or no, wait, no, it's right here. Oh, I see. Oh. Okay, so we have a bunch of graphs. So for each subject, oops, this guy got shifted over. For each subject, we'll have, what is this, 5 graphs. So you get one graph of the raw data, right, unfiltered. One graph of just RMS. One graph of moving average. And then one graph with the raw data with the moving average overlaid on top of it, and one for the raw data with the RMS overlaid on it. And I have this so that you can see what the filtering does to the data, because it's filtering. So it will actually, like, it can change what it looks like, right? Like, this is the raw data, this is RMS, this is the average. So why would you need to filter it? It's because, like, as you can see here, like, this data is like scratchy, right? Like, it dips down a lot. The reason why it does is every time they blink, your pupil dilation drops to 0. So every single blink has a drop down to 0. And so why you would use RMS versus moving average is it's so to get rid of those, like, spikes and kind of clean up the data a bit. It's still kind of noisy, but, like, it's better than nothing. So here we have, and this is with 90% overlap and a half a second window. You can play around with the overlap and the window to try to, like, get different, to see what looks better. I didn't really play around with a whole bunch. But the less overlap there is, it just changes some stuff. It'll look a bit different. So that's why it's important to look at the overlay, right? So this, you can see it's mostly the same, right? It mostly just chops off all of this, like, down, this noise down here, right? All these, like, 0 spikes, because it's averaging it out. So why would you use root mean square versus moving average? Root mean square cuts out more spike, right? Like, this goes down, right? Down here, this is like 35. Well, this one, I would say this is, oh, it's about the same. Sometimes it's better at chopping off the spikes. Like, see this one, like these peak down here, right? This goes all the way to like 15, while this one is like up here in like 25-ish area. So RMS is better at making it more, at getting rid of those long spikes all the way down compared to the moving average. They are both valid ways of smoothing it, though. So that is sort of a personal preference, or like a lab call that you guys might want to talk about what exactly you want to use and why. They have different, like, reasons why you'd want to do either. And so you might also see that there are these black bars, right? These are each, there's one for each slide, right? So this is Cahill gray, light bulb gray, rat gray, right? This is the, there's one for each slide. So basically divides it up in 2 slides. And so you can see like the Cahill is the longest one, and it's the first one, and it continues on throughout the, basically slice it up so you can see maybe if there are trends, right? Maybe you see, like, there's a spike here, there's a spike here, there's a spike here. I don't know what that means, but maybe that each of these slides, they might be the scary ones. I don't know. I haven't looked at this. Something to do in the future. Teaching you how to use this code. So yeah, so it'll produce graphs, all these graphs for all of this data, and some of it will look better than others. Like, this guy is kind of gross looking, right? Like, look at this. It's also spiky. I don't know what's going on here, but maybe it's an error. That's why you got to look at the eye tracking code to see like what's error and what's not. But those are all the graphs. And if you want to close all of the graphs, all you need to do is type close all down here and press enter, and it will close all of the graphs, right? They're all closed now. That's just a quick trick on how to close all the graphs. And then that is about it for the, yeah, that's basically all you need to know for the full analysis. Basically, the full analysis just graphs the data. That's why you would use it. That's why you want to use it. If you want to figure out graphs, mostly I was using this so that if you wanted to see general like trends or something, it would be easier to, it's basically to visualize all the data. That's why we do that. I would imagine usually you would just do order it by slides and do the partial analysis. That's probably enough for most of what you guys want to do, but I want to give you some flexibility of what to choose. I'm trying to think if there's anything left, but I think that is it for the most part. So thank you all for listening, and hopefully that was helpful for how to use it. And good luck.